L. Courtney. I grew up in a family that went fishing a lot, so I'm really familiar with lakes, ponds, the ocean, and fish. When I was in 6th grade, I was homeschooled, and as part of my science, I weighed and measured every single fish that we caught. I became really familiar with the relationship between weight and length in fish. I also learned there's an equation to predict the weight of a fish based on its length, the weight of a healthy fish. The thing is, this equation has some errors. The coefficient doesn't have any real physical meaning, and this lack of a physical meaning has led to errors in widely used and trusted sources about what a healthy fish weighs. But more importantly, this model yields parameters with large uncertainties, so you're not sure about the expected weight of a fish. In 2011, I was part of a small study to develop an improved model, an improved equation that didn't have these problems. The thing was, I only used 5 species of fish, and peer reviewers said this is not enough to propose an improved model. So in this study, I used a lot more species, over 94 taxa and 14,000 individual fish, and I proposed an improved weight length model for fish in order to improve bioindicators of ecosystem condition. So what is a bioindicator? A bioindicator is any feature of an environment that you can measure and that you know changes based on how healthy the environment is. It's like a canary in a coal mine. Miners would send down a canary into a coal mine, and if there were even trace amounts of carbon monoxide or methane, which would kill the miners, the canary would die, and they would know that the environment wasn't safe. In the same way, fish serve as a sentinel of aquatic environment health. If the fish and environment aren't healthy, nothing is healthy because fish are so interconnected in the food web. So how do you tell if a fish is healthy? Well, the equation that I talked about gives you the expected weight, the expected healthy weight of a fish of a given species. So you compare the measured weight of a fish to its healthy weight in something called condition index, where you divide the measured by the expected weight, and condition index is what you use as a bioindicator to tell how healthy it is. Expected weight is traditionally found, the equation I talked about, by A times L to the B, where A and B are parameters you get by fitting data from a population to this model. The thing is, this coefficient A doesn't have any clear physical meaning, so when you do a fit, there's no gut reaction, that it never looks wrong. And this has led to errors in widely used sources like the Carlander Handbook of Freshwater Fisheries Biology and fishbase.org, which lots of people use to define a healthy fish. But if they're wrong, that's dangerous. More importantly, the traditional model yields large uncertainties, in especially the A parameter, but both parameters, which leads to limited bioindicator accuracy. If you're not sure what a healthy fish should weigh, how can you be sure if the environment's healthy? So this research is really important because if an improved model could predict expected weights with better accuracy, it would lead to more accurate assessment of ecosystem condition. Fish have been successfully used as bioindicators of hazardous metal levels, water quality by the Environmental Protection Agency, coral reef health, oil spill impact, and the effects of invasive species. The 2 bottom pictures here are locations where original data was gathered, and on the upper right you see a Gulf Manhattan with a cranial tumor that actually came from Calcasieu Estuary, and the cranial tumor serves as a bioindicator of metal contaminants in this area. This is not from my study, this is from a different study, but it just goes to show that fish are accurate bioindicators of the health of the entire ecosystem, not just fish. So this study proposed an improved model. Weight in terms of length. So the healthy weight of a fish of length L is equal to 1,000 times length over L1 to the B. So instead of multiplying by A, I divide by L1. The cool thing is, L1 now has a real physical meaning. L1 is the length of a healthy fish that weighs 1,000 grams or a kilogram. This concrete meaning makes it easier to catch errors because L1 should be about 450 millimeters. And if it's really far from that, you should double check your results to make sure you don't cause hazardous errors. The primary hypothesis here was that this proposed improved model would provide significantly smaller uncertainties for both parameters for 2 different types of fits to the traditional model, and therefore be able to provide more accurate bioindicators of ecosystem condition. I also had a secondary hypothesis that this new parameter I introduced, L1, since it has a physical meaning, would be able to accurately diagnose environmental issues in systems already known to be stressed, so we're just testing it. We're not trying to do original research. I'm testing whether L1 is an accurate and sensitive bioindicator. In order to assess these problems, I used 160 data sets encompassing more than 90 species of fish and over 14,000 individual fish. 41 of these were originally measured data sets by my family and this encompassed more than 2,000 fish. The other data sets were gathered from the Colorado Department of Parks and Wildlife and the Carlander Handbook of Freshwater Fisheries Biology, and they were carefully selected to ensure lots of different body types and lengths and weights of fish to make sure that this improved model would be accurate no matter what type of fish or what body type it had. So I used everything from the American eel and the brook lamprey to sturgeon and gar, which can reach 20 feet long, to make sure the model is applicable for every kind of fish. So then I had to test the model once I had all these data sets. For each of the 160 data sets, weight versus length data was plotted on a graph. Then, each of those 160 was fit 3 times, to the improved model and then the traditional model, with both a nonlinear least squares and a linear least squares fit, because scientists differ about which one of these provides more accurate parameters, and I wanted to cover my bases and make sure that if the improved model is better, it's better no matter what. The thing is, these 3 fits yield different parameters. They yield L1 and B, A and B, and a different A and B. So I had to transform all these to be equivalent so I could actually compare the results. I transformed them from the nonlinear least squares fit using L1 equivalent equals 1000 over A to the 1 over B. I did a similar thing for linear least squares fit. I also had to propagate the uncertainties to be equivalent to L1 for A equivalent, and B, because the point of the whole thing is to compare uncertainties and make sure it lowered uncertainties. After I had done all of the 480 fits, I graphed. So this is one of the 480 fits that I did. This is length versus weight in cutthroat trout from 11 Mile Reservoir. Each dot represents a fish, and the red line represents the fit to the improved model. This is an example of the output from the Cy-Davis program I used for graphing, where L1 and B are given, and their uncertainties are listed as well. So this is what it looks like for each of the 480 data sets. After I did this, I graphed the results. This shows all the uncertainties from all of the 480 fits for the improved model, traditional model, and the other traditional model fit. The green X's are the errors in the improved model, and they are clearly the lowest. They're reliably lower, they're clearly lower in almost every case compared to both traditional model sets. If you'll notice, this is a logarithmic scale to the left, so it's not evenly spaced from 0 to 100. The improved model uncertainties hover around 1%, whereas the traditional model has uncertainties anywhere from above 1% to 100%. The improved model clearly reduced L1 uncertainties and had them lowest in the majority of cases. This looks good, right? The thing is, the uncertainties in B were not dramatically reduced. They were simply comparable in every case. But thankfully, since B uncertainties were comparable and L1 uncertainties were greatly reduced, the improved model is clearly better than the traditional model. Especially because L1 uncertainty directly impacts the expected weight and condition factor. The uncertainty in expected weight is at least that of A or L1, so it's really important to minimize A or L1. When you minimize them, like I did in the improved model, you get more accurate bioindicators, which means that you can detect problems in environments with less samples of fish, and you can detect problems earlier in emergence. You can address them before they get out of control. So this means that the improved model is empirically better than the traditional model. It produces more accurate expected weights for bioindicators, and parameter L1 has a concrete physical meaning, which makes errors like those in the Carlander Handbook and fishbase.org easier to catch. The secondary hypothesis was that the parameter L1 would be useful itself as a bioindicator. Now remember, L1 means the length of a healthy fish that weighs 1,000 grams or 1 kilogram. So if a fish has a longer L1, it's skinnier than a fish with a shorter L1 because it's longer to weigh 1 kilogram. Skinnier fish are more unhealthy than heavier fish. So the way you use L1 as a bioindicator is to look at the average L1 for a population of a fish in an area. And so say it's 450. Now, if that same species of fish has an L1 of 500 in another area, it's less healthy because it's so much skinnier. Heavier fish have an earlier maturity rate, higher reproductive rates, and have more energy stored so they can go longer between feedings. A heavier fish is indisputably a healthier fish. So if an L1 goes up in a population, it shows that the fish has gotten more unhealthy. The way I tested if L1 was a good bioindicator was to look at 3 systems that are known to have problems and see if L1 was able to detect these problems. The first system I looked at was Calcasieu Estuary, Louisiana. I looked at 2012 original data versus 2000 statewide Louisiana averages. I looked at red drum, black drum, and spotted sea trout. And after the Deepwater Horizon oil spill, many areas were closed to oyster harvesting. The Calcasieu was left open, so it experienced extreme overharvesting of oysters and the damage of a lot of valuable oyster reef habitat. As you can see, for all 3 species, L1 saw an increase, which means that all 3 species got less healthy. Black drum were hit the heaviest, with about a 6% increase, which actually corresponds to about 18% underweight, which is a big deal. So why were black drum hit the hardest and red drum and spotted sea trout not hit as badly? This is because black drum are bottom dwellers, and they eat oysters. They're a benthic species, and oysters are one of their biggest foods. Red drum and spotted sea trout live higher in the water column, so they were affected a lot by the reef damage and the environment damage in general, but were hit less hard because they don't rely on oysters like black drum do. So L1 was able to pinpoint the problem in this environment, so that's a positive step towards it being a good bioindicator. The second environment I looked at was Lafourche inshore waters in Louisiana. This, I was looking at the effects of the oil spill itself and the oil in the water on the same three species. So L1 showed that red drum and spotted sea trout saw a decrease in health, but black drum did not. Is this right, or is L1 failing as a bioindicator? No, this is right. Scientists have concluded that the heavier oil had already sank to the bottom before reaching Lafourche inshore waters here. So the black drum, being bottom dwellers, had nice clear water and weren't affected by this. But the lighter oil was still up in the water column, and so it did have a negative impact on red drum and spotted sea trout. So L1 was able to not only diagnose environmental problems, but to pinpoint the areas of the water column that were affected by this oil. So it's successful in 2 out of 3 cases so far. The third case, I looked at a completely different environment. I looked at Blue Mesa Reservoir in Colorado. In Blue Mesa, kokanee salmon and rainbow trout are native species, but lake trout and brown trout are invasive species that became extremely overpopulated in the last few years. They have decimated the kokanee salmon because all 3 of these species eat kokanee salmon, which is a problem. It worked out fine. There was only rainbows and kokanees, right? They balance each other out. But these species caused a severe underpopulation in kokanees. So the trout species all experienced a decrease in body condition, with the brown trout being almost 36% underweight. That's horrible. But the kokanee salmons are healthier? Does that make sense, or is L1 wrong this time? No, it's right also, because since the kokanee salmon population was decimated, there are fewer kokanees, but their food source was fine. So each individual kokanee got more food. So each individual kokanee salmon is actually healthier. So in 3 out of 3 cases, L1 has proved to accurately show the problems in the environment. So I propose that L1 is a sensitive bioindicator, and because its uncertainty is lower than the traditional model, it should be used in conjunction with condition factor to indicate ecosystem health, because it'll pinpoint problems earlier and with fewer samples, so it's all around better. Now back to the primary hypothesis, the improved model. A healthy weight for a fish of length L is equal to 1,000 times L over L1 to the B should be adopted as a new weight length model for fish, because it produces more accurate parameters than the traditional model does, and therefore will provide more accurate bioindicators for ecosystem condition.